Hi, this is lesson 31 in the use of JASP for understanding statistics. In the late 1970s, Doug Kenrick, a professor that was at Arizona State when I was a student there, had conducted a study where he found that individuals who were exposed to nude centerfold pictures, particularly male participants who were exposed to nude centerfold pictures, subsequently would judge the attractiveness of their romantic partners as lower than if they viewed neutral stimuli of some sort. So in more recent years, someone tried to replicate those results, and they used the following 2x2 design that is shown up here. They had roughly half and half females and males, and they displayed to them either abstract art or pictures of attractive nudes. And they looked to see how they would respond in terms of rating the pleasantness of those as a manipulation check. How much do they like viewing the nudes or the art? And he also looked to see whether or not he could get the replication to show that the rated attractiveness of their romantic partners would go down. Let's take a look at those results. If you open up JASP, go Open, Data Library, go to ANOVA, and Erotic Pictures in Love. Takes a bit to load. I want to get rid of all of the instructional materials that have been pre-run here and just get the data set. Okay. One thing I want to do is to make clear the levels of the variable. You'll notice that JASP does not insist that you have numbers. It's my habit to put numbers in for variables like condition or gender and then to label them, but you don't have to. If you double click to open up the data mode, I'm going to put in for labels 1 for female and 2 for male, just to make explicit for learning purposes. In the label, close the window, apply, that we have 2 levels that can be numbered 1 and 2. I'm going to do the same thing for condition, so double left click, go to the variable labels and put in 1 for abstract art and 2 for nudes. Click someplace else and apply, and now we're all set up. Let's go to our analysis. We're going to go to analysis, then go to ANOVA. Classical ANOVA. And let's start off with our manipulation check. So our fixed factors are independent variables. We have 2 independent variables. We're going to use condition as factor A and gender as factor B. We're going to start off looking at the dependent variable of average pleasantness to see if the manipulation worked. So, did males judge the pleasantness of the nudes higher than the males? This was something that Doug Kenrick didn't test. So the follow-up researchers wanted to look at how the manipulation was perceived. What we see, if you look at this condition, is factor A. Gender is factor B. And then the A times B interaction is here. Remember, they do not show the between groups overall. They don't show the total. They show the residuals, and that's your within group variance. The F for condition is less than 1. So it can't be significant, and the p-value supports that. It is much greater than 0.05, non-significant. Gender, the f-value is large with 1 and 219 degrees of freedom. F with 1 and 219 degrees of freedom is 10.185. You're generally going to find F criticals of around 3.2, 3.4, somewhere in that range for significance. And so that's way over, and the p-value is definitely less than 0.05. For the interaction, with an F of 1 and 219 degrees of freedom, you have an F value of 22.29, which again is significant, and it's less than even, the probability of making a type 1 error is less than 0.001. Now, what do these effects mean? One thing to look at is your descriptive statistics. These give you your cell means and the number of people in each of the means to help you interpret what is going on. So if these are cell means, let me go ahead and map that on for you with my Excel program. And I'm going to put in means for females abstract art 4.904. Males abstract art is next, and that is 4.658. For those seeing nudes, for the female, that was 4.201. And for the male seeing nudes, it was 5.473. Okay. Now, what I would teach in my undergraduate classes and graduate classes is that you can get marginal means by taking the averages. So if I take the averages of those two numbers and the average here of those two numbers, I see very little difference on the whole between abstract art and nudes. They're both averaging out to be about 4.8. The average there is 4.55. And the average here is 5.065. Here you have one of the larger differences, although not as large as this difference right here. So males overall look like they are finding it more pleasant, and that is consistent with this main effect for gender. You have a main effect for gender, that means you can interpret the difference between these two means. You don't have a main effect for condition, which means that you have to assume that you shouldn't interpret the difference between these two marginal means for factor A. There is a significant interaction, and so that means you can interpret the cell means for a difference. Now, the best way to see this is to go to descriptive plots, and let's put condition on the horizontal axis and gender as separate lines. Grab this and pull it out, and it becomes nice and neat. And here it becomes obvious that for the males, the pleasantness they ascribe to looking at the nudes is higher than that for abstract art. For females, it appears to be the opposing effect, where they have a decrease in their pleasantness at looking at nudes compared to the abstract art. So it's going in the opposite direction. Now, let's look at marginal means. I already calculated some marginal means, but if you're just working from the computer, what you would do is look here and do the marginal means. It gives you the cell mean, so you can see that kind of broken down to help you interpret it, but it also gives you the averages. And are those averages the same as what I calculated? Let's take a look. For abstract art, 4.871. For nudes, 4.837. Yes, those are the same. And for the females, 4.553 and males 5.06. So as you get used to looking at the output, what you learn is that if there's no significant main effect for gender, you don't interpret those means. There is a significant effect for gender, you do interpret these marginal means. And if there's an interaction effect, you look to see how these are different. Okay, those are your basic interpretations. There's a little bit further you could go, but I'm going to call it there for simplicity's sake, except I want to look at the other variable. Did we actually replicate the results? This is the results for just the manipulation check. I'm going to take average pleasantness out of the equation. I'm going to look at partner attractiveness. And when we look at the graph, I'm going to pull it out so I can see it. Now, notice all of our F values are less than 1. If the F values are less than 1, there's absolutely no significant effect to be found. This is supported by our p-values. None of them are less than 0.05. They have to be less than 0.05 in order to be statistically significant. So none of these F obtained values surpass the F critical values. There's no main effect for condition. There's no main effect for gender. There's no interaction effect. When you look at these means, the graph, it looks like there's a pattern such that the lines are not perfectly parallel, but they're not significantly deviating from being parallel. And you can see this a little bit better if you just go over to the left and you click on error bars and you look at the error bars to see where the means are expected to be on the perceived partner attractiveness. And what we see is there's a lot of error. So the true mean for females could be anywhere around here. The true mean for males could be anywhere between here. There's a 95% chance that it's somewhere in between there. Same thing over here. You have a lot of sampling error. So those true means could be in a lot of places. And one of those places is they could be such that, for example, if you drop this mean down a little bit over here, the lines become parallel. So they're not significantly dissimilar from a parallel structure, so there's no significant interaction. You can't just look at the graph. The graph is useful and important for interpreting a significant interaction, but the graph in of itself is uninformative as to whether or not there's anything significant. You use the F test to determine if there is a significant interaction. So the researcher, Bazarini, I don't know how to pronounce that, in 2017 did not replicate the effects. Is that because of some difference in the experimental procedure? Is it due to changes from the 1970s to 2017? I don't know, You can go read the articles, and I believe some of those factors, possible explanations are there, or maybe this effect was never there. Um, Hard to know, but the effect did not replicate. So that's the two-factor analysis of variance, and that's how it maps on to our classroom taught structure. See you next time.